All right, so we're changing gears a little bit. So Sue was just talking about system suitabilities and tracking system suitability. I'm going to take a little bit of time to talk about publishing data with Panorama. So after I introduce you to Panorama, we'll loop back around and talk a little bit more about QC and system suitability, because Panorama has some built-in system suitability AutoQC functionalities. Then we're going to loop back around and talk more about the publishing data with Panorama. So we're going to kind of be ping-ponging back and forth for a little bit. So first, just what is Panorama? PanoramaWeb.org is a website that you can go to that stores all of your Skyline documents. So kind of the idea was we were, as a community, pouring all of our time and effort into making these Skyline documents, right? So we would develop these methods, we would refine our methods, we would validate the methods, we would do all of this data integration and data processing and annotate these files, and then they would just die on our desktops in our labs. So the idea was we should build a place to share these things so that everybody can learn from other laboratory science. So it's a public repository. You can also keep projects private. And we have 3, I think, Panorama partners in the pharmaceutical industry that have their own private servers, so they can all access each other's data, but none of us can see what they're doing. You can start a project in Panorama, keeping it private while you're working on it, and then you can, for example, give reviewers, when you're ready to submit a manuscript, you can give them a key so they can log in and see your data. And then after the paper gets accepted, you can make it go public. You can make that project go public. So if you were to do something like that, authors and other submitters would only be able to look at your document, but you could, for example, be making changes as editors, as peer reviewers might be giving you suggestions or asking you to double check certain things or collect more data. You could be making those changes. They will only be able to see it. And then finally, speaking of publishing, if you're looking to publish in journals like Molecular Cellular Proteomics, MCP, they have publishing guidelines for targeted mass spectrometry that Panorama and Skyline will satisfy. So you'll be able to get past some of those submission requirements too. So I hopefully have sold you now on this idea of PanoramaWeb. How exactly do you do it? The first step is to make a folder in Panorama, and I believe Nat is the guy that would help you set up an account and everything with Panorama. So spam his inbox with requests. You would set up your Skyline document. You would use Skyline just like we've been teaching you in the course. Then you would import the document, and I'll show you in a little bit how we can do that, into the folder. And then you can also associate all of your raw data and upload those raw data in there too, because in step 4, you can get a proteome exchange ID for your raw data too, which also we need to share our data for publishing in mass spectrometry. So kind of a one-stop shop for getting all of your data sharing requirements. Step 2, you annotate the, not in the Skyline document annotations, but you annotate that Panorama project. So you have to add some certain things like for this Panorama project and this set of Skyline documents, what the abstract would be for that project, details about the experiment and how you prepared the samples, which mass spectrometer you used. You would hit button submit, and then you get a proteome exchange ID for all of those raw data. You can get a shortened URL for your project. So instead of having like some garbity like set of numbers and letters, you can just have it be called like mypaper.url. It's really nice for reproducibility and transparency. So I have talked to a couple of you now about like, oh, but like looking at the data, like what if the data looks like junk? Like what if my chromatograms just look terrible or my spectra just look terrible? when you use Panorama in this way as a community, not necessarily like you yourself have to upload all your data, but you can use other people's data. And maybe someone makes a claim in a paper and you're like, I don't really know if that peptide or that protein is really regulated differentially. You can look at their chromatograms and make that judgment yourself. So the first step, when you go to upload these Skyline documents, I added in little screenshots up here for you. But if you looked at your Skyline document, and here I'm just using like those calibration curve data files that we had been doing before, there's a button up at the top, 1, 2, 3, 4 icons in, right there, right next to the save. Right next to the save, the little floppy disk icon, there's the publish up to Panorama. Once you have a Panorama folder set up for a project, that's all you have to do to push your Skyline document to Panorama. You'll get this little web page for your Skyline document on panoramaweb.org. And you would have whatever your project's going to be called, and then you would have all of your Skyline documents associated with it. So just the 4 steps: you upload, you annotate, you submit, and then that's it. So it's really, it took me a little bit to get sold on the idea, but once I did it the first time, it seems really, really simple. Once you get your project in there, the annotations, you just add a title for whatever your project is, the organism, the instrument. If you did any spike in, it's just a checkbox. So if you use internal standards, you would just check the box. The experimental details, once you have the paper, you can associate the paper. So the citations, if anybody were to use your data off of Panorama, you can very easily have them cite you. The submit step is literally just a button called submit. There's nothing tricky about it. You can get your little shortened URL. So instead of this being some long string of alphanumeric characters, you can just call it CPTAC Study 9. So if you were to go to this address, you would see all the data for the calibration curve stuff that we were doing yesterday. And then finally, all the raw data, when we publish raw data in mass spectrometry, we're supposed to make it publicly available, the actual like .raw files. So when you put those raw files in the Panorama folder, they'll be assigned proteome exchange ID that you can put in your paper. So you've got all of your Skyline documents. For MCP, you've got your raw data, proteome exchange ID. Everything is kind of just done for you. So that's kind of, I think, everything I had wanted to say. When you submit it submit your paper and you still want to keep it private, your project, you can see everything, but you can make that read-only copy, so your reviewers would still be able to see it, and it would still be kind of the same link, so you don't have to worry about that little short URL accidentally going public, even if somebody else were to share it. Yeah. Are there any questions about kind of these steps for Panorama? Because next I'm going to point you to Panorama. We're going to do a little hands-on.
If someone were to, how exactly can you explain how exactly the submission is the submission from Panorama go directly to proteome exchange?
Yeah, so all of your raw data files are in the Panorama folder. And then when you hit submit, there's like a couple annotations that you have to do for Panorama. That's like that organism and the mass spectrometer that it was collected on. And then when you hit the submit button, there's an option to get assigned a proteome exchange ID. Okay. Yeah. You don't have to do it. Hypothetically, this person wouldn't have to do anything. Somebody else is going to do it for you. Yeah. Oh, yeah. 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 Yes.
I have just a more general question. Suppose we wanted to data mine the entirety of how well does PanoramaWeb support that?
So Panorama is mostly targeted experiments. So as far as data mining, the computational lab that I'm a member of mostly uses Massive and Pride repositories for doing data mining. It's a lot of DDA experiments in there. Raw files for targeted experiments are maybe not fantastic for data mining.
You have a really nice format where you said things about whether or not they're in journals and that sort of thing. And so if you're like trying to understand how a field constructs itself,
Yeah.
it's a nice data set. So
Yeah.
yeah, I was just curious if you're set up for automatically script mining it.
We have, so I'm going to point you guys to go to this URL. And I've only picked this one because I collected the data myself, so I'm very familiar with how it was set up. So we're in panoramaweb.org/singlepointcal.url. And I'm going to go there too, I think. .org. Ta-da! You should get a page like this. So this is an example of a Panorama Web project that I had initially started, and it was closed. It was just for me to see. And then the paper went out for review. So the reviewers got the special access to it to look at it all. If I log in, you'll notice in the top corner, the sign in, if I were to log in, I would get a whole bunch of options to edit this page. So I'm just going to show you that real quick and hopefully have remembered my sign in. So now I'm going to have some things in here that you're not going to be able to see because you guys all have read access, but I have edit access. So I could go up here and change all kinds of things in this document. I'm not going to because I don't want to break it. I have all of the annotations in here. So I have a short little abstract describing what my Panorama project is. I have a description of how I generated the samples and generated the data. You can see I used two different types of mass spectrometer. In this particular project, I used a Q-exactive and I used an Altus. I didn't have any spike-ins. I've got my proteome exchange ID. If we were to go to the data pipeline, you'll show I'm not doing any kind of data analysis in here. I did everything in Skyline. I didn't have to do anything while I was in Panorama. Everything I did in Skyline was performed prior to uploading this to Panorama. If we click on the raw data tab, you can see I've got organized in here my SRM files from the Altus. And you can see all of my raw files have been uploaded in here. For some reason, I decided I was going to include solvent blanks too, if you're curious about my solvent blank situation. You can see the Q-exactive files. If we go back to the Panorama dashboard and go down to this yeast Osmo SRM and click on that, you'll see this is an SRM experiment. So this was a targeted experiment. I targeted 71 proteins comprising 140 peptides, 140 precursors, 645 transitions. I have all the proteins that I targeted are shown. You can see the peptides that were targeted for those proteins. You can see that they were not heavy spike-in. You can see their mass to charge. And you can see their chromatograms. So luckily, I happened to pick one where the chromatogram looked good. That would have been embarrassing if I picked a bad one. So this is my solvent blank sample. There is no peptide in the solvent blank sample. But you can see the chromatogram just like it would have looked in Skyline. But you can view all of these right in Panorama. You can see summary charts just like in Skyline. You can see the peak area. You can see the retention time. If I scroll all the way back up and go back to that yeast osmo. Other things that we can do in here is if I go up one more level, I think. Yeah. So you'll notice there's the download here. If you click that, you'll download the whole Skyline file, and you can just open it up in Skyline, which can be kind of nice. If you wanted to look at DIA data, I have the exact same samples acquired data independent acquisition style. I think this is everything I wanted to show you about Panorama project. Are there any questions about this part so far? Yes. Yeah, I did not have any problem. I have not only the raw files, but the converted MZMLs. And it took some time to upload, as you might imagine. But other than that, I did not encounter any limits yet.
Okay, that's a good question. I'm not much familiar with DDA, but if you already have everything on Panorama, do you even need a proteome exchange ID?
For the raw data needs to have a proteome exchange ID. So Panorama. Yeah. Well, it's just like if you submit to like Pride or something, you'll get like a Pride identifier and a proteome exchange. So instead of like Panorama doesn't have like a Pride ID kind of thing. You just have like the URL. Well, yeah, because the Panorama ID is really just for like the Skyline documents. The raw data needs to have an identifier too for the raw data set. Yeah. Before, what we were doing was uploading all of our data, all of our Skyline data into Panorama, and then uploading our raw data into like Massive or something, and then having that identifier and then this identifier. So this was just a way to try to like contain everything instead of having to split it between my Skyline document and my raw data. Okay. So now, while we're here, I'm going to show you Panorama Public, and maybe this gets at the data mining question. So if I just click up on the Panorama logo, it'll take me to the front page of Panorama. If you scroll down, there's a big blue button, Panorama Public. Hit that. So this is where everybody's public Panorama projects live. So if you scroll down, you'll see like all the various Panorama Public projects. So we've got all kinds of craziness. We've got some GUI ducks. We've got some, I don't even know what this is, oysters it looks like. We've got some humans, some E. coli. You can see the publications. You'll see that some of these do have publications associated with them. But some people haven't even published yet, but have already made their data public. So huge props for them. You'll see here's my single point. But let's go up, and at the top, you'll see there's, for example, protein search. So let's say I was interested in ApoA1. I can search all of the Panorama projects to see if anybody has ever made an experiment for ApoA1. And you'll see, wow, a lot of people seem to have done ApoA1. So now if I were, for example, starting that plasma tutorial, one of the proteins I think I asked you to try to develop an assay for was ApoA1. If I didn't know what of all the very many peptides I wanted to target in ApoA1, maybe for prior knowledge, I would go to Panorama and just ask, has anybody ever seen ApoA1? I could click on what looks like a good one. Analytical protein standards. Sounds good. I'll click on that. Oh, the Prego manuscript. Nice. All right. I can go in here and click on, oop, sorry, I clicked the wrong thing. I'm going to click on, I think I want ApoA1. There we go. So I clicked on the wrong thing first if you were following me. So on the first one, instead of clicking analytical protein standards, click on the protein. It'll take you straight to the protein. So in this particular project, they searched, they were targeting ApoA1. So this is showing you the whole protein sequence for ApoA1. And then these boxes are showing you the peptides that they had in their document that they detected. So for this particular protein in this particular manuscript, they actually got like pretty decent sequence coverage for ApoA1, which makes sense because in this particular project, they made protein standards for all of the proteins that they were targeting. So this was like a purified protein standard that they made just by in vitro producing a plasmid version of the protein. So they got a lot of coverage, but now maybe I want to say which one of these peptides was the best. Here's all the peptides that they found for it. And now I can start looking at chromatograms. This one looks pretty great. If I were developing an assay for ApoA1, this looks like it would be great. We can see all of the peptides at once with all of their different peak areas. So these are all of the peptides that were detected for ApoA1. If I were developing an assay, maybe I would start with this guy because he's the most intense, but maybe I would pick like 3 or 4. So this might get closer to the question of data mining. I don't think we have any scrapers, web scrapers for this yet, but if you want to build one, that'd be great. Yeah.
There is actually MRM SADB, can go to PanoramaWeb.
Oh, can it? MRM Assay DB is a scraper for Panorama? Nice. So you don't even have to build your own scraper. Yes.
So in a case like this, is there any guarantee that those peptides are stereotypic or unique for that protein?
There is no test in Panorama for whether they're unique or not. Yeah.
So yeah, buyer beware.
Mm.
For a peptide, is there no requirements technically from these kind of data sets that those peptides that are selected are to that protein? So it might be a family or something like that. So it's always a good idea to do a lab search yourself to make sure that you want to see if it came from protein.
Mm-hmm. Yeah. Yeah. So that's Panorama Public. And I think I might have time before the break. Oh, yeah. So this is in the Prego manuscript. So it's got all the publication information, and there's not a like straight email me here contact on here. Well, I think you can still see who it was because when it gets uploaded, there is a name associated. Not sure where to find it now. Yeah, I'm not seeing an easy way to find it, but I know there has to be because you can see who uploaded stuff. So I'm gonna have to look for that during the break. Yeah, that was kind of a very brief introduction to Panorama Public for your mass spectrometry projects. It's really nice because I'm a huge believer in the open data and transparency, especially as proteomics is getting bigger and bigger data. We have the potential to be putting out more and more garbage. And as you start getting into the statistics portion of this program, I think it's important to remember that your statistics can only work as well as your data is. Like if you have garbage data, you're going to get garbage out, right? So it's nice to, if you're looking at your data, decide for yourself, is the data of high enough quality or not? There's only really the 3 components you need to publish that project. You need the Skyline document, the raw files, and to know something about the experiment and how the data was generated. But it's overall pretty nice to have everything in one-stop shop. Any questions on Panorama in general? Okay. So I just showed you Panorama for projects that are like experiments, but we can also use Panorama QC. Now, we just got done with Sue's lecture telling us that QC and system suitability are not the same. It was a very, very unfortunate naming choice when they decided to call it Panorama QC, because everything I'm going to be talking to you about is really Panorama system suitability. So that was unfortunate naming. One of the nicest tools that I've been introduced to in the past few years is AutoQC. Again, in your head, replace QC with system suitability. So AutoQC is a small tool that runs on your instrument computer, and it will see that you are collecting system suitability data. It'll upload it into a Skyline document, process it for you, push it up to Panorama, and perform S-Pro-Cop statistics. You do nothing. You just run the actual acquisition queue. So it's been really nice because before, how I was doing system suitability was I would queue the system suitability acquisition, then I would upload it into Skyline, then I would assess it myself, and then I would just keep doing that indefinitely forever. Now I can just log into Panorama and see whether or not my system is still in spec with S-Pro-Cop statistics. Setting up AutoQC is pretty straightforward if you have your own instrument. So this is, again, just that front page of Panorama. On the side over here, there's, under Documentation tab, there's a tutorial called Quality Control with AutoQC. So it has all of the details for it. And we are just going to go there really quick. So I'm going back to Panorama. I'm just back on the main website of Panorama. Under Documentation, Quality Control with AutoQC. So it's got the 2 download links. You have to match your AutoQC loader with whichever version of Skyline you have installed on your instrument computer. So if your instrument computer is using Skyline regular release, download AutoQC loader. If you're using Skyline daily on your instrument computer, download daily. So the workflow does a really good job. The tutorial does a really good job of telling you how exactly to set up. But I want to show you just what it looks like. So when I open up AutoQC Loader on my instrument computer, we have a whole bunch of user profiles, because the way we have set it up in Mike McCoss's lab is each user on each instrument has their own personal kind of, not like standards, but our own kind of like file organization. We are currently working on everyone doing the same file organization, but we have, for example, Lindsay, Brian, Mike, Sandy, and when I log in, all I do is check my profile, and then that's it. I open up, I get my column equilibrating, I check my profile in AutoQC Loader, and then I just do everything like I would normally do. To set up that configuration, looks something like this. So if I were a new user on AutoQC, I would set up the file path to my Skyline document. My Skyline document will have whatever my system suitability peptides are in it. So if I'm using like the biagnosis peptides we were using before, I would have a Skyline document with just those biagnosis peptides, just like how we set it up when we were going to do IRT. The folder to watch is wherever in your file naming scheme you're going to be saving your QCs. So my folder to watch, I have it in our instrument. It's like data, and then it's Pino, and then under Pino I have like my data folder. So I say watch the data folder under Pino, and then the file filter, I name all of my system suitabilities with the same tag of like system suitability. So all of my acquisition files that are system suitability, I write system suitability, and I say file filter, look for any system suitability named file. You can set that. Yeah. So you can have it set to ping as far back as 30 days. So I usually just leave this default, and then it checks after each acquisition. So it's like once an hour, basically. It'll check that folder. You can set the acquisition duration. So I know some people doing small molecule have very, very short gradients. So they decrease this to reflect whatever their acquisition time is. So like if their acquisitions are like 5 minutes, they would decrease this expected acquisition duration to 5 minutes to increase the ping on that folder. Does that make sense? Yeah. Your instrument.
Yes.
Your instrument computer, not your like personal desktop.
And it's also not possible if you direct it to the folder to watch, there's actually on that computer, or that's the one that's where everything's loaded.
Everything, the Skyline document, the AutoQC loader, and like Excalibur for like actually queuing your instrument, all needs to be on the same folder. Yeah. Yeah.
If I have like several instruments, can I have several profiles checked in parallel?
Yeah, well, because do you have one computer for several instruments?
No, but I have several instruments mapped on my computer. Like I have, actually, I have like my computer, my LCMS system somewhere else, but I have the data folder where the QC is coming in, mapped on my computer, and this is where the AutoQC also is.
Yeah, then I guess that's where it would have to be. I guess in my lab, maybe it's set up weird. Every instrument we have has its own computer to run the instrument. So that's where we put our AutoQC. So each one of our instruments, that Altus and that QExactive, each have their own computer. They each have AutoQC, and I have a profile on each of them. Does that make sense? Yeah.
I think, I don't know, for us, the instrument computers typically don't have internet. So,
yeah, if you don't have internet, you would not be able to push up to Panorama, but you could still use AutoQC. It would just stay local in a Skyline document.
I mean, what Roman was explaining, just you can then have a network folder on your laptop then, which just, I mean, if it works,
It's
yeah,
definitely
can be
possible
out
to
there.
have AutoQC on another computer and it would.
Yeah, yeah. The way we have it set up, we just put AutoQC on each of our instrument computers, but it sounds like your lab might be set up differently. Yeah.
If you're mapping to a network drive, then there be separate directories for each unit.
Yeah.
Yes. You
Mm.
would just put that in the path.
The question was whether I can check in from several profiles, watch several folders.
You can have multiple profiles checked. We only ever have one checked at a time, though. Yeah, I think it would probably it probably would work. I can't imagine why it wouldn't if you had multiple checked.
So I wonder, as a separate option, if I had one LCMS that was capable of doing nano and high flow, so I had two separate data sets, both on high flow, whatever I
Mhm.
follow, but that could be two separate profiles.
Yep, I would have two separate profiles for that, and I would probably have two separate Skyline documents for it. Yeah. I think that was the last thing I wanted to show. Oh, no, never mind. So if you are connected to the internet and you want it to push to Panorama, you can sign in to Panorama, and this will link it to auto push that Skyline document. And I'll show you what that looks like after the break. So now we'll kind of like pretend we're all set up now for AutoQC, and when we come back from the break, I'll show you what it looks like in Panorama.